Good morning, good afternoon, good evening from wherever you are. Welcome to the What's Next in NanoEHS webinar. I am Jay Vitas, Branch Chief of the Emerging Technologies Branch, National Institute of Occupational Safety and Health, also known as NIOSH, and co-manager of the NIOSH Nanotechnology Research Center, and I will be the moderator for this event. The NanoEHS webinar series is an important platform for agencies participating in the National Nanotechnology Initiative, or the NNI, to share information on federal nanotechnology, environmental, health, and safety research progress and findings. Before I introduce the panel, I will share some background on the webinar theme and how it fits in with the priority NanoEHS activities coordinated by the NNI. I'm going to share some good information here as well. First, I want to note that the U.S. government's commitment to understanding the potential health and environmental implications of nanotechnology has been a central part of the nanotechnology research framework since the early days of the NNI. The creation of the Interagency Nanotechnology Environmental and Health Implications Working Group, also known as NEHI, under the NNI was pivotal in charting a path forward to address key NanoEHS research questions. NEHI is spearheading the review of the NNI's current EHS research strategy, which was published in 2011. The updated strategy, or updated draft strategy, describes the substantial progress since the strategy was published. It also points to needs, questions, and directions to meet the NNI's goals of responsible development of nanotechnology. The draft strategy has been released for public comment through July 22nd of 2024, and included here on the page that I've shared with you is a QR code and a link to the regulations.gov page where you can view the document. We welcome and look forward to your comments on the draft. Please share this request for comment with your colleagues and collaborators. Okay. The NNI public webinars have been an important avenue for connecting diverse NanoEHS experts with the public. The series continues to share perspectives on the progress that had been made in understanding EHS implications of engineering nanomaterials, the NNI's role in answering these research questions in each of the NNI's core areas. As the NNI approaches the 21st anniversary of the initiative's authorization, we will continue to focus on rising experts from academia, industry, and the U.S. government on future directions. Today, we are excited to bring together two researchers leading the next phase of NanoEHS research. We will hear from Brian Zhang and Nathan Drew. Brian is a MITAX fellow and a member of the Vireo Advisors team. He is a toxicologist and a postdoctoral fellow at the University of Alberta. Brian's research includes work on a safer by design toolbox for early pre-commercial toxicity and risk screening of functionalized forms of cellulose. Nathan's a statistician in NIOSH's Emerging Technologies Branch. His research includes using grouping frameworks to derive occupational exposure limits for nanomaterials and evaluating data needs for potency estimates in quantitative risk assessment. Now, before I turn it over to Brian, a couple of housekeeping items. Each of the presenters will provide their presentation followed by a few minutes of questions and answers. You're welcome to add questions at any time using the Q&A tab at the bottom of your screen. If we don't get to your question, we will do our best to follow up with you to obtain an answer. Finally, I hope that you will join us for some of our other webinars in this series. You can find information on the NNI public webinars on our website, which is very easy to remember, nano.gov. And now, over to Brian.
Okay. Thank you very much for the introduction. So I'm Brian. Good afternoon and good morning, everyone. So today I'm going to talk about the recent progress in evaluating the environmental health and safety of cellulose nanomaterials or nanocellulose. So I'm going to start with my presentation talking about the types of cellulose nanomaterials used in commercial applications, and then discuss toxicity studies on different types of cellulose nanomaterials and the challenges we are facing. I will end this talk with an overview of Safer by Design toolbox and the life cycle risk assessment we have been developing for nanocellulose. So there are 4 common types of nanocellulose used in commercial applications. So the first one is cellulose nanocrystal, so CNC. The second one is temple oxidized cellulose nanofibrils, so TCNF. So those both are rod shaped with low aspect ratio. The third one is CNF, and the last one is microfibrillated cellulose, so MFC, which has the most complex morphologies with web-like network structure. So cellulose nanomaterials have been used in a wide range of applications, such as cosmetics, automotive components, food contact materials, and food uses. And one of the emerging applications in food use is scaffold for culture meat. This is largely due to their ability to improve the tensile strength of the material and reduce water vapor and oxygen permeability. Cellulose nanomaterials are also biodegradable and biocompatible, making them ideal candidates as sustainable material to replace plastics and also used in medical products. So with so many applications, so the safety and the transparency in the applications is crucial for pre-commercial assessment. However, we are facing a lot of challenges of EHS assessment of such material. So characterizing a material is a fundamental first step in any EHS assessment. It is also essential to understand how similar it is to other forms of cellulose, and the types of EHS considerations are required. However, there is a lack of standardized testing protocols for characterization, especially for CNF and MFC. For example, what is a proper dispersion technique we can use? Vortex, sonication, high shear mixing, or something else, right? Because the dispersion not only affects physical chemical characterization, but also affects their potential toxicity. As for toxicity testing, we have limited standardized testing protocol specific for nanocellulose, and it's difficult to track such material in biological and environmental system. And it's also difficult to assess the environmental fate, such as biotransformation and bioaccumulation. And so cellulose is derived from nature and is one of the most abundant natural material that has been used in food supply for a very long time. So even though we expect materials to behave similarly to conventional cellulose, but we still need to proactively to demonstrate the safety. So this is the work our group has been leading. Next, so let's look at some toxicity studies conducted in recent years to help fill in the data gaps in the safety of cellulose nanomaterials. So the first one is the oral toxicity testing on SD rats. So the animals were fed with either soccer flock, which is a conventional cellulose, CNC or CNF, up to 4% of their diet. Clinical observations listed below were recorded during the experiment. 7-day, 14-day, and 90-day oral toxicity testing suggests that no adverse effect associated with any of the exposure, indicating CNC and CNF behave similarly to conventional cellulose, and there is no safety concerns when used as food ingredient up to 4% of the diet. However, we are still facing some challenges of performing the oral toxicity testing. First is administration options is limited to oral gavage and dietary only. Secondly, cellulose is a component of almost all the diets, so it is not recommended to use a fiber-free control. Instead, using conventional cellulose as control group, which as we use soccer flock in the study. And we also need to use representative forms expected to be used in food, not just any forms of the cellulose. And often we need to consider additional endpoints in testing than what is recommended in the standardized testing. So absorption, distribution, metabolism, and excretion. So ADME study is designed to investigate how a chemical is processed by a living organism. So this can be achieved by labeling the chemical of interest. To track nanocellulose, we can use first the fluorescent labeling, which is stable and have relatively low toxicity. However, it's difficult to quantitatively measure that in the biological fluids and the tissue, and the fluorescent label may alter the behavior of the material and thus change their toxicity. So this study used fluorescent labeling as a technique for ecotoxicity and environmental fate study. So in this study, zebrafish larvae at 166-hour post-fertilization were exposed to either 100 milligram per liter Texas red labeled CNF in picture A or Texas red alone. And the picture C shows the presence of Texas red labeled CNF in the esophagus of the zebrafish larvae, indicating uptake of CNF by the zebrafish larvae. As for the toxicity testing, zebrafish embryos were exposed up to 100 milligram per liter CNFs between 24 and 120-hour post-fertilization. There was no mobility was observed in any of the treatment, including CNF and Texas red labeled CNF and Texas red alone at any given concentrations at any observation times. So the second way to track nanocellulose is using radioisotope labeling, which has much higher sensitivity compared to the conventional fluorescent labeling. However, it is expensive to synthesize radioisotope compound, and the experiment can be quite time-consuming. And there are some additional challenges of using radioisotope labeling to track nanocellulose. So first is the presence of residual sugars in the cellulose nanomaterials, which can be labeled during the process. So it is very important to have washing steps to remove all the residual sugars to ensure only exposure is to the cellulose. The second challenge is the presence of free carbon-14 label. For example, we conduct an experiment on carbon-14 labeled fibrillary cellulose. We measure the radioactivity of the C14 labeled cellulose and the radioactivity of the filtrate of the filtered C14 labeled cellulose. And the results shows we have measurable level of radioactivity in the filtrate, indicating the presence of unbound free C14 label. So currently we are working to conduct an ADME study using C14 labeled MFC. So to evaluate the biokinetics of MFC and see if it is similar to conventional cellulose. So historically, researchers gained information about toxicity of substance through animal testing. However, these studies can take months or even years to complete and can be quite expensive. So in recent years, more and more companies are interested in avoiding animal testing to gain entry in markets such as cosmetics, and many regulators are committed to reducing and replacing animal testing with new approach methodologies. So the Dr. Chris Isaias from Baylor University, in collaboration with various advisors, have developed a variety of models for safety assessment, including gastrointestinal model for simulated oral exposure, alveolar lung model, dermal and epidermal cells model. So these models are part of the Safer by Design toolbox to evaluate the safety of next generation cellulose nanomaterials. And this Safer by Design toolbox project is funded by P3 Nano and the US Endowment. So the NAM studies provide methods and safety database to develop this toolbox. In addition, physical chemical characterization and methods provides database for different chemistry and forms of cellulose nanomaterials. This toolbox also includes the SOPs for physical chemical characterization of pristine nanocellulose, as well as their properties in biologically relevant media. So Dr. Yulo Xu from UC Davis synthesized 4 different temper oxidized cellulose nanofibrils, so TCNFs, using sodium hypochlorite with different concentration at different blending time. And this is how we name our materials. We also have 4 different prior-date chloride oxidized CNFs, so PCCNFs, and softated CNFs. So the physical and chemical database for all these forms are included in the Safer by Design toolbox. So here I'm going to provide an example of NAM study using simulated gastrointestinal digestion model, aka gut on a chip model. So on the top, this image shows the simulation of oral exposure in human. And on the bottom is the gut on a chip model in the intestine. It consists of 2 channels separated by a permeable membrane. Various cell types such as mucus layer, epithelial cells, and immune cells can be seated on both sides of the membrane for exposure experiment. So in this study, we used CACO2 and H229 cultures. They were exposed to different CNFs for 4 hours. The barrier integrity was measured by using trans-epithelial endothelial resistance, so TER. And this graph shows the TER 8 days post-exposure. So the positive control had a significant reduction in the resistance. 1 SCNF, 2 TCNFs, and 1 PCCNF had a significantly lower TER over 8 days post-exposure as compared to untreated cells. However, they are not significantly different from the vehicle control. We also tested the effects of CNFs on a GI triculture model, including cell death, inflammation, and oxidative stress. And the study shows no adverse effect associated with exposure, suggesting cellulose nanomaterials behave similarly to conventional cellulose and no adverse effects in gastrointestinal model. So this Safer by Design toolbox is bringing together pieces of project to fill in the data gaps in physical chemical characterization and the safety of cellulose nanomaterials for the evaluation of the cellulose nanomaterial through life cycle risk assessment, so LCRA. So LCRA describes the life cycle of a particular product from the raw material all the way to the end of life by developing quantitative exposure scenarios that describes the potential exposure impacting workers, public, and the environment based on intended use. These scenarios are ranked by applying exposure criteria, and the risk assumption uses simplified worst-case scenarios. So next, I'm going to briefly discuss life cycle risk assessment of SCNF and TCNF for food packaging applications to demonstrate the safety of priority commercial cellulose nanomaterials forms and applications. So the first step of LCRA is to identify the entire life cycle of a particular product from raw material to the end of life. For each life cycle stage, possible scenarios that could lead to occupational, public, or environmental exposure are identified. We also identify likely exposure routes. So following the scenario development, scenarios are ranked based on the hazard and exposure by applying exposure criteria, including directness of the exposure, meaning how easily are particles released, magnitude, which relates to the relative size of exposure based on percentage of the CNF. The third one is likelihood, which prioritizes intentional exposure over unintentional or accidental exposures. And the last one is the frequency. It is an estimate of how often an exposure is expected. So this table here includes an example of our case study showing the highest ranking scenarios. So following this exposure scenario development and the ranking, the next step in the LCRA is to prioritize exposure and hazard assessment on the highest ranking scenarios. So that's just a brief overview of life cycle risk assessment. So to sum up my presentation, so the cellulose nanomaterials showed little to no toxicity in both in vivo and in vitro studies. However, there are still a lot of challenges and gaps remain in material characterization, ADME studies, chronic exposure, and etc. And the safety, then the safer by design toolbox can be applied to select safe forms of cellulose nanomaterials for commercial applications. And at last, I'd like to acknowledge the funding agency, the U.S. Endowment of Forestry and Communities, and the USDA, U.S. Forest Service, Forest Product Laboratory, the partner of the Alliance for the Food Safety Acceptance of Fibrillate and Crystalline Cellulose, Matex Elevates, and the National Nanotechnology Initiative provide this opportunity for me to present our project. And the last but not least, the amazing Vario team, their contribution on the project and their help with the presentation. And thank you for your patience, and I would like to answer questions.
All right, thank you so much, Brian, for your presentation. And to the group, please, I invite you to ask any questions of Brian using the Q&A tab at the bottom of your screen. I'll start out with a question for you. You mentioned two methods to track nanocellulose for ADME studies. What approaches are used to evaluate the potential for cellular uptake of nanocellulose?
Yeah, so what we typically use a fluorescent labeling, right? So we can label nanocellulose with different dyes, and then we can image those under confocal microscopy. One thing we need to pay attention to is we need to use 3D reconstruction of the image to confirm if the nanocellulose is inside the cell. Because most of the time, if we only have the 2D image, maybe we have a just example, we have a 2D image and we have a fluorescent in the middle inside the cell. It looks they are inside the cell, but if you change it to the 3D reconstruction, it may be on the surface of the membrane, so they are not inside the cell, they're just on the surface. So we need to use a 3D reconstruction to confirm that those fluorescent is actually inside the cell within the cell membrane.
No, excellent. Thank you.
Sorry, just something I want and also it's very tricky to do fluorescent labeling because we have autofluorescent, and sometimes I mentioned there are some free labeling. So which means we need a control group to to control those, to confirm there is no autofluorescent or no free labeling in the samples.
Another question for you from the audience. Can cellulose nanoparticles be used as a potential replacement of highly cytotoxic silver nanoparticles?
Ooh, yeah, it's hard. Like, actually, I don't know the answer for that, but so here's my guess. So first, it depends on which nanocellulose we are talking about. because the morphology varies and the size distribution varies between just CNC and all the way to the like CNF, MFC. And also it's depending on what's the commercial application you are looking for.
But thank you. Another question, in the risk assessment table, are the columns weighted equally in terms of classifying low, medium, or high? Or is one column more important than the other?
The table has so the low, so we mean low, medium, high. This is just I can go back to that. Can I just share again? So in the okay, so all those are weighted equally. So we have 3 different levels, and we have different cutoff. For example, the magnitude, if it's low, it's lower than 1%. If it's a medium, it's 1% to 10%. And if it's a high, it's over 10%.
Okay. No, thank you very much, Brian. Okay. Well, I've continued to add in. We may have time at the end to bring Brian back on to answer additional questions, but for now, I'm going to turn it over to Nathan Drew. And please pull up your slides and share your great work with all of us. Thank you.
Thanks, Jay. Thanks again, Jay, for the introduction. I'm Nathan Drew, and today I will be describing an approach where we're trying to develop. We're developing an approach, that is, rather, to develop occupational exposure limits for groups of engineered nanomaterials. And to begin, I have to acknowledge my NIOSH colleagues, Eileen Kempel and Teresa Boots, because they've been invaluable contributors to this effort. So the motivation for this effort is that historically, an occupational exposure limit has been for a specific substance, and it requires data from at least a subchronic rodent bioassay. And these types of studies are expensive and they're time intensive. This leads to the fact that many substances, including engineered nanomaterials, will lack an occupational exposure limit. So currently, the 21st century goals for toxicology call for the utilization of alternative assays and methods to better utilize resources and also reduce animal use. So a categorical occupational exposure limit is a concept for developing group-based occupational exposure limits for materials with similar hazard for a given adverse health effect. And this can align with those 21st century goals. So in the process of developing occupational exposure limits for engineered nanomaterials, one of the first steps is just to see, is there already an authoritative OEL for that material? Probably not, but it makes sense to check for that first. Next, it makes sense to see, could the data that are available be used to derive an occupational exposure band? And this is guidance published by NIOSH in 2019. It's going to have some requirements, such as one requirement may be that it needs to be at least a 28-day inhalation exposure. So if that data is available, then an occupational exposure band could be derived. But again, that is a potentially stringent data requirement. So then we move into step 3, which is to explore alternative methods. So there are a lot of these, such as read across. These can make use of shorter term data. And what we'll show here is one such approach where we get into hazard potency grouping. So this framework that we use consists of 5 steps. So first, the necessary data must be collected for the materials of interest. We need the physicochemical properties for those materials, and we need to have responses for our endpoints of interest. The second step would be to estimate the hazard of those materials, and we go about that using benchmark dose modeling. The third step is when we have those initial hazard estimates, we want to identify groups of materials with similar hazard estimates. So I'll describe two approaches to identify those groups of similar materials. And the fourth step, that's where we develop a classification model. And the goal there is to think about what if we were presented with a new engineered nanomaterial, what might we know about it to predict its initial hazard group? And the idea there is that it likely will not have toxicological data, but it probably has some physicochemical information. So the classification model will take in those physicochemical properties and output a predicted group. And then lastly, once that new material has been classified, it's made a hazard group prediction, then an initial estimate of hazard could be generated. So for the first step in obtaining the data that we used here were collated from within NIOSH, from NIOSH's external partners, and mostly from systematic literature searches. I won't get into the searches here, but the details available in the Boots publication listed from 2021. So the focus of these searches were on rodent toxicology studies of acute pulmonary inflammation. This endpoint was selected because lung inflammation is on the adverse outcome pathway for occupational lung diseases and inhaled particles. And acute lung inflammation is a commonly reported endpoint in rodent toxicology studies. And again, that aligns with that 21st century goal. From all of these studies, the data that we extracted were information on the dose response associations, information about the experimental design, such as the route of exposure, the species and strain of the subjects, and then lastly, the physicochemical properties of the materials used within those studies. So now we have this pool of data, and our next goal is to estimate the hazard of all of the different materials that we have. And again, to go about estimating the hazard, we used benchmark dose modeling. And the goal is to estimate the lung dose associated with an increase of 4% in the neutrophilic response. And this is shown to be biologically relevant. So it's a good value to estimate. Across the data sets and the publications that we selected, the experimental designs varied, as you may expect, such as by the different species, by the different routes of exposure. So to get them all on the same page, we took the administered doses and converted those to a deposited dose, which is normalized by lung weight. So that's in micrograms per gram lung. The modeling was conducted using the US EPA benchmark dose software, following sort of the standard guidance there. And as a result, what we ended up with were 116 benchmark dose estimates. Now, a part of this is that essentially what we did is we had to cut down the data that we initially selected, because to do benchmark dose modeling, what you need is at least 3 exposure groups, so that includes the control, and there needs to be a statistically significant trend. So if a study didn't meet that kind of bare minimum requirement, then at this point we were unable to use it further. So we have the data, we have our initial estimates of hazard for these materials. So then the next step is to start figuring out what materials have similar hazard estimates. What are the groups of materials that we can use going forward? And we approached this using 2 different methods. The first method we used was agglomerative hierarchical clustering. And what's nice about this approach is that it's flexible and it allows for some exploration around how many groups may be sufficient for this data. So in this approach, similarity between materials and their hazard estimates is defined as the Euclidean distance between their hazard estimates. And then as the agglomeration is conducted, basically, as the clusters are then combined in future steps, that was determined using Ward's method for the linkage. The second approach that we considered was assigning these materials to predefined bins based on orders of magnitude. The nice thing about this approach, on the other hand, compared to agglomerative hierarchical clustering, is that these are a little more intuitive. The band definitions tell you what those hazard estimates are. Additionally, this approach is robust in the sense that as data could be added in the future, these bins don't change. Versus the agglomerative hierarchical clustering, those are data driven. So as the data changes, those clusters may change. So when looking at the what might be the right number or a good number of clusters for the agglomerative hierarchical clustering process, one tool that can be used is the scree plot shown here. And what's being measured is essentially the density of the cluster. How close are the hazard estimates to one another? So it starts to level off fairly quickly. There's some evidence that 4 or more groups is probably sufficient, as the densities doesn't really change much after that point. And what we found is that really what seems to do the job for dividing up these materials is to use 8 hierarchical clusters. What this chart on the right is showing, so the points, so each line represents the hazard estimates for a given material. The benchmark dose of that material is to the right, and then to the left is the lower confidence limit on that benchmark dose estimate. So most of these materials, the largest proportion, were assigned to cluster number 1, which is at the bottom. That's the most hazardous group. And that was about 38% of these materials. And then it kind of divvies up from there. And as you can see at the top, we didn't have a lot of materials in the least hazardous groups of 7 and 8, but there were still a few of those. For the order of magnitude bands, since these were predefined, for these data, 7 orders of magnitude were sufficient to capture the range of the hazard estimates. And in this approach, 45% of materials, again, that's the largest proportion, were assigned to the 10 to 100 micrograms per gram lung band. All right, so again, kind of stepping back and thinking about what we have the data, we've developed the initial hazard estimates, we have initial groups of materials with similar hazard. So the next step is, again, to start thinking about, well, what if we had a new material? What would we do? How would we come up with this initial hazard estimate? So the approach that we took is we wanted to develop a classification model to predict the hazard group. And again, the idea here is that this new material probably will not have toxicological information, but it should have some knowledge about its physicochemical properties. So our goal is to now predict either the hierarchical cluster or the order of magnitude band for a new material using only its physicochemical information. And for these data, we extracted 21 different properties. To make the classifications, the approach that we chose was a random forest. So what this process does is it takes in the physicochemical information, it's input into a large number of different classification trees that each make a vote, and then those votes are tallied across the forest, and the output is a predicted hazard group for that new material. So again, the input to this model are the physicochemical properties, and what these are going to be, what these represent are those that are commonly reported in the published literature. We were basically taking what we could get, as much as we could get. Unfortunately, these properties are not always reported, nor are they always reported in the same way. And in this analysis of these 21 properties, there was significant sparsity, I would say. So for 12 of the properties, that information was available for less than half of the 116 materials. There were 4 properties that were always available, and then 5 properties that were available for at least half the time. Not all of the time, but more than 50%. And in developing the random forest model, I'll try not to get too technical, but for the most part, we stuck with the default choices. So in this case, this random forest consists of 500 different classification trees. But in the development of the model and the tuning of some of the parameters, we used 10-fold cross-validation, which was repeated 10 times. So this tuned one of the parameters that essentially chooses, as these trees are being created, how many of the physical chemical properties will be considered to create that given tree. And also this process was used to identify the final model. For both of these approaches, the hierarchical clustering model and the order of magnitude band, the final model is evaluated by its out-of-bag performance. All right. So in both of these cases, what we found is that these final models, again, they're either predicting the hierarchical cluster or they're predicting the order of magnitude band. Both of them perform better than a naive model. So what I mean by a naive model will be one that just simply always classifies a new material into the largest group. It needs no other information. It's just always going to pick the biggest group. So for the agglomerative hierarchical clustering model, in that case, the largest group was cluster number 1. 38% of the materials were in that group. So in that case, the naive model would be accurate 38% of the time. But this random forest model, which utilizes the physical chemical information, was accurate 47% of the time. And that was statistically significantly higher than the naive model. Similarly, for the model that was predicting the order of magnitude bands, there the largest group was the 10 to 100 microgram per gram lung band, which was 45% of materials. But again, the random forest model that makes use of the physical chemical properties was able to be accurate 63% of the time. And again, that was statistically significantly higher than that naive model. Now, both of these models do a pretty good job. They were more accurate than a naive model, but both models did misclassify some of the materials. So for example, both of these models would place a hazardous material into a lower hazard group about 1/5 of the time. So they're certainly not perfect, but they do a good job. And looking more into the random forest models and kind of what they're doing, how they're working, how they're making decisions. For the model that was predicting the hierarchical clusters, what we found is that the core material was the most important predictor of the hazard group. So what I mean by that is it a titanium dioxide? Is it a cerium oxide? Is it a multi-wall carbon nanotube? Just knowing that sort of base substance, the core material, was the most useful predictor. After that, what we found were the most important predictors of the hierarchical cluster were the primary particle characteristics. So describing the material size. So things like specific surface area or the diameter. Those were also key predictors. We found similar results for the random forest model that was predicting the order of magnitude band. Again, the most important predictor was the core material. And between these two approaches, really the top 10 roughly predictors are similar between the two, but the rankings do vary a little bit. So as an example of kind of how this process might work, what's kind of going on with this random forest model, suppose that we're presented with a new material and all that we know is that it's a titanium dioxide. So in this data that the models were built on, titanium dioxide was one of the more commonly reported materials, and it was present in 7 of the 8 hierarchical clusters. For these data, the largest proportions of TiO2s were present in clusters 1 and 3, so among the higher hazards. So if that's all that we knew about this material, all that we knew is that it was a TiO2, we'd have to make a decision. Should we initially assign it to cluster 1 or 3? And maybe if we're taking a precautionary approach, we could assign it to cluster number 1. So as we start to learn more about the physical chemical properties and we input those into the model, these cluster assignments can and probably will change. So for example, if we also know the scale of that new titanium dioxide, and if it happens to be microscale, that's going to change the cluster assignment to cluster number 5. So that's going to lower the hazard cluster. But if we know that this is a nanoscale TiO2, that would remain in cluster number 1. And as a last example, and this would continue across all of the different PChem properties, suppose that we know that we have a nanoscale TiO2, and we also know something about its shape. So for example, if this nano TiO2 is spherical, then that will lead to the assignment in cluster number 3, reducing the hazard group a little bit. But if it's fiber-like, such as a nanobelt, then that would remain in cluster number 1. So in conclusion, I think what this illustrates is that we have an approach that can predict a new material's hazard group using only its physicochemical information. And while this approach focused on acute lung inflammation, this could be pretty directly expanded to other endpoints or assays. Once a new material's hazard group has been predicted, this could be used to develop an initial categorical occupational exposure limit, or it could be used as an input into another tool. And lastly, I think this approach could be improved, and I think a large source of improvement could be accomplished by further collaborative efforts, such as sharing data and not relying on what's published in the literature, and also standardizing data, because as we discussed, those physical chemical properties, if those were more standardized and more commonly reported, that would likely lead to a better model. So some quick references. And with that, thanks for your attention, and thanks to the NNI for this opportunity.
Great. Thank you so much, Nathan, for your presentation. To the audience, this is your time. Go ahead and throw in some of your questions, both for Nathan and his presentation, which we'll start out with first. And then if you have any lingering questions of Brian, you're welcome to throw those in the chat as well. And I know Brian's been answering some of those good questions that people are asking me, so I really appreciate your engagement. I'll start out with a question for you, Nathan. Could the model that you presented today be used to predict an initial hazard group for nanocellulose?
No, not this current model. And the reason being is that this model has not seen acute lung inflammation data for nanocellulose. But if that data were available, then these initial groupings and the model itself could be updated.
Oh, that's great. Yeah, thank you. One question talks about what kind of tools, other tools you might have tried, and references AutoML. I assume that's a machine learning tool. Have you tried the AutoML tool, and potentially would that have provided a better model?
I'm not familiar with AutoML. I will say this is not the one method to rule them all. There are other ways to classify. There are other classification models, and those could be explored. I think, random forests have been shown to be a pretty good off-the-shelf baseline model to use, and we were able to get good performance out of it. But there's certainly opportunity to plug in whatever other classification models might be useful.
Yeah, one of our more questions for you. One of our audience members asks about when this will be published, but I'm going to say, as being part of your group, that we are still working to get that published soon. But then I'm going to throw the more important questions to you. Will NIOSH use this method to propose recommended exposure limits for common nanomaterials in commerce that do not have specific RELs, such as various graphene-based materials?
Yeah, so I can kind of talk about both of those. In terms of publication, there's an earlier version of this framework that was published in 2017 in Regulatory Tox and Pharmacology. And then an external review draft of this report was published on the Federal Register in 2021. And then, like Jay said, it's currently being we're trying to wrap it up and get it out to the next step. For the second question, do we plan to use this tool to develop RELs for new materials in commerce? Eventually, yes. But something to keep in mind with the approach in the model shown here is that this was for acute lung inflammation. So again, kind of thinking about historically what's done to develop an occupational exposure limit, that's going to be at least a subchronic, so 13-week or longer rodent bioassay. A lot of the data used in this approach were intratracheal installations, so basically a single exposure. So this approach at this time could not be used to develop a REL because there are uncertainties about how to go from these acute exposures to thinking about a working lifetime value.
Okay, another question. For the categorical occupational exposure prediction, is it possible to have a base set of properties to avoid underestimating hazard?
Maybe. So I think the reason I say maybe is that it's, you know, we came up and I showed those initial estimates of how important these predictors were. But again, I think it's important to keep in mind that most of these properties were missing for most of the materials. So before that question, I think, could really be tackled, there needs to be, we need to identify opportunities to start to fill out these physicochemical properties. Maybe there's a core set that should always be reported, and then that information would probably be useful for the initial group predictions.
No, thanks, Nathan. I'm going to go back to one of the questions set here. We were talking about whether or not the NIOSH might use these to propose recommended exposure limits. The follow-on question to that is, will regulatory agencies accept those results from this clustering procedure to predict chronic worker exposure inhalation limits?
Good question. So certainly any type of process like this one is going to need to be validated. And what that's going to require, again, is ensuring that the data that these models are trained on is representative. So for this approach, these materials are not representative of all engineered nanomaterials. What's in the published literature that we were able to obtain, we have a lot of titanium dioxides, we have a lot of zinc oxide, we have a lot of carbon nanotubes. Like the first question, we don't have cellulose. So as new materials come out, this model would probably not do a good job at making a prediction for a novel material because it's not been trained to do so. So at this point, no, I don't think there would be acceptance for these models, particularly this model, because there needs to be a lot more work done to validate them.
Thanks, Nathan. All right, I'm going to bring Brian back up on the screen, and welcome again any questions to both of our fantastic presenters today. I do have one question, Brian, that regards, are there currently any safety data sheets regarding nanocellulose? And welcome your answer to that question.
You're muted.
Yeah, you're the challenge of coming off mute after being on mute for so long. Thanks, Brian.
So regarding the nanocellulose, does that mean the nanocellulose we synthesized or any nanocellulose in particular? So if the nanocellulose is commercially available, they have safety data sheets.
Oh, great. Awesome. That's fantastic. See here, there's also a question, are there any in vitro assays for interaction with macrophages that could inform interpretation of pulmonary tox in vivo assays?
Good question. Yeah, and
Yeah, it's good.
was that
Go ahead, either one, go ahead,
okay?
Nate.
I'll go first.
Go
Sorry.
ahead.
Yeah, so that is another potential future effort for this approach is how can we best incorporate other alternative assays? So if we do know something about a new material's in vitro toxicity, can that and how could that be incorporated into this scheme? I think that that information can be incorporated, but that is a future step.
Yeah, Brian, I welcome your response to that question as well, because I think you're both sort of working different angles of the same issue.
Yeah, so for us, we're actually right now we're conducting and collecting data for the in vitro toxicity testing on the lung exposure and for using including the response of macrophages. Yeah, but we're still collecting data, so I can't give the answer right now.
Great. One more for you, Nathan. Would the 21 PChem properties change for chronic worker recommended exposure level predictions versus the acute inflammation predictors?
That's an interesting question. I'm trying to look back at the PChem properties to see if there's something that may not be applicable in a chronic assay. I don't think so. You know, again, I don't want to make it seem like these 21 properties were special in any ways. These were what were commonly reported. These are what's in the published literature. We may not need all of these. We might need additional ones. So it would really depend, I think, on what's available from those for the materials that have been used in a chronic exposure. There's more work to determine which physical chemical properties are most useful at predicting hazard group because they're so sparse.
No, great. Awesome. Well, thank you both for some wonderful presentations today, getting the juices flowing on where we're going next, really with respect to risk assessment and methods and some of the challenges associated, particularly with engineered nanomaterials. And I think you did a great job of both presenting some of that today. Well, of course, I also want to thank all of you out there for your excellent questions, for participating in this particular webinar, and for tuning in. If you're interested in our series of webinars, please, again, visit our NNI public webinar page, the simple nano.gov, where you can find recordings of these presentations as well. You can also follow us on X, formerly known as Twitter, at NNI Nano News. And so again, appreciate everyone's time and participation, and thank you very much for our wonderful presentation today. And join us next time.